Good morning. Let's discuss on cholesterol chemistry and metabolism. Cholesterol is the principal sterol of all the higher animals distributed in the body tissues, especially in the brain and spinal cord. Cholesterol is a major sterol in the humans and it is not synthesized in the plant. Cholesterol is the amphipathic lipid which can be synthesized by most cells of the body, the all nucleated cells of the body, and is obtained from the diet of in the foods of animal origin. The major sources of dietary cholesterol is egg yolk, very rich source. The yellow content of the egg contains huge amount of cholesterol, dietary products, and meat, and particularly liver. An abnormally high concentration of cholesterol in the blood is is a risk factor for atherosclerosis that can lead to myocardial infarction or strokes. And cholesterol is a component of cell plasma membrane and is required for transport across the cell membrane. It has a very important role. It has given a very important peculiar texture to the plasma membrane, that is the semi-fluidic nature. So that is all because of the semi-rigid nature of the cholesterol. Chemically, it is the carbon C27H46O, and it has 27 carbons. At the third position, there is a hydroxy group, and there is a double bond between 5 and 6 carbon. And then there are two methyl groups at the junction of two rings at C18 and C19 position. Then in the side chain, there is an R8 carbon chain. So this is the structure of cholesterol. Cholesterol has to perform versatile functions in the body, and the four major function of this steroid is the production of bile salts, production of vitamin D. It is also responsible for synthesis of all the six hormones, or steroidal hormones can be synthesized from the cholesterol. And biosynthesis of cholesterol happens almost all the nucleated cells of the body at slower or higher rate, but the main sites for synthesis of cholesterol are liver, intestine, adrenal cortex, ovary, and testes. So how it is being synthesized? The two major precursor for synthesis of cholesterol are acetyl coenzyme A and NADPH. So the source of acetyl coenzyme A, right from glycolysis from glucose to pyruvate, and pyruvate can be converted into the acetyl coenzyme A. This acetyl coenzyme A in the mitochondria, so it converts to citrate with the help of oxaloacetate. This citrate comes out in the cytoplasm, and it can be broken up into the acetyl coenzyme A. So this is one important source for cholesterol synthesis. Then secondarily, this NADPH can be synthesized mainly from the HMP shunt. HMP is the main source for production of NADPH and pentose sugars. Then malic shuttle is also another source for production of NADPH. Then cytosolic isocitrate dehydrogenase enzyme, they also lead to the synthesis of this NADPH. So these are the three important sources for generation of NADPH. So this NADPH acetyl coenzyme A, they are responsible for production of cholesterol, and this requires the investment of ATPs. So there are five major stages of cholesterol biosynthesis. There are step one is formation of HMG coenzyme A, then formation of mevalonate, conversion of mevalonate to isopentenyl pyrophosphate, then formation of squalene, then conversion of squalene to cholesterol. So here is a tentative, steps given. In between, there are so many steps, but five major steps are compiled here. Right from acetyl coenzyme A, it converts up into HMG coenzyme A, and from HMG coenzyme A to mevalonate, mevalonate to isoprene unit, that is isopentenyl pyrophosphate. Then squalene is there, and then lanosterol, and finally cholesterol. In between, so many steps are there. we will see later on. The two major stages takes place in the cytoplasm, and the next three stages take place in the endoplasmic reticulum. Like the synthesis of HMG coenzyme A and mevalonate are happening in the endoplasmic reticulum of the they are happening in the cytoplasm, and remaining stages are happening in the endoplasmic reticulum. So the major synthetic pathway is like it starts from two acetylene coenzyme A molecules. These two molecules fuse with each other in the presence of acetoacetyl coenzyme A synthase enzyme, and this leads to formation of acetoacetyl coenzyme A. Then another in step 2, another molecule of acetyl coenzyme A fuses and leading to the formation of beta hydroxy beta methyl glutaryl coenzyme A, and the enzyme is the HMG coenzyme A synthase. In the step 3, that is the rate limiting and major rate controlling step, that is the HMG coenzyme A reductase enzyme. And in this step, this HMG coenzyme A further converts up into the mevalonate, which is the 6 carbon species. And then 2 NADPH are consumed here, leading to the formation of 2 NADP. Then mevalonate further, after 3 consecutive kinases reaction, 1, 2nd, and 3rd, it finally converted up into the 5 pyrophosphomevalonate, which is also a 6 carbon. Further, there is a release of carbon dioxide in the presence of decarboxylase. It leads to the formation of third important product intermediate, that is the isopentenyl pyrophosphate. And this isopentenyl pyrophosphate, or IPP, with the help of enzyme isomerase, is converted up into the dimethyl allyl pyrophosphate, which is also 5 carbon species. Further, there is another molecule of 1 isopentenyl pyrophosphate fuses in the presence of enzyme transferase, leading to the 10 carbon species, that is the geranyl pyrophosphate. And this geranyl pyrophosphate again fuses with another molecule of isopentenyl pyrophosphate and leading to the formation of 15 carbon intermediate, that is the farnesyl pyrophosphate. So there are 3 consecutive addition of 5 carbon species in the form of isopentenyl pyrophosphate, and they finally lead to the formation of farnesyl. Further, 15 carbon farnesyl condenses with another molecule of farnesyl pyrophosphate in the presence of enzyme squalene synthase, and here also 1 NADPH is consumed, and leading to the formation of squalene, that is the 30 carbon precursor of cholesterol. Further, epoxidase enzyme converts this squalene to squalene epoxide. Further, cyclase convert this squalene epoxide into the lanosterol, which is again 30 carbon. In step 7A, there is a consequent release of 3 carbon atom, loss of 3 carbon atom in the form of carbon dioxide. Finally, it is converted up into the gymosterol, which is a 27 carbon. From gymosterol to desmosterol, that is also a 27 carbon containing intermediate. And further, in step 7C, it is finally converted up into the cholesterol. So these are the 7 major steps of cholesterol synthesis. Regulation of cholesterol. As I have already told you, HMG coenzyme A reductase is the main regulatory rate limiting enzyme of this pathway, because this is the slowest step. So all rate is dependent on this. This is being regulated at the level of hormones by the two important hormones. One is the insulin, thyroxine, which are favoring the synthesis of cholesterol, while the glucagon is disfavoring the synthesis of cholesterol. So insulin and thyroxine, they lead to the dephosphorylation of HMG coenzyme A reductase, which is converted up into the active form. And in this active form, this leads to the synthesis of cholesterol, while in the phosphorylated state, which is being facilitated by glucagon, it becomes inactive and it inhibits the synthesis of cholesterol. Second important way is through covalent modification of the enzyme, and third is through feedback regulation, which happens at the genetic level, decreasing the transcription of gene for synthesis of HMG coenzyme A reductase. So excessive amount or sufficient amount of cholesterol will inhibit at the level of transcription of gene and inhibit the synthesis of HMG coenzyme A reductase. Then metabolic fate of cholesterol. What are the uses? How it is being converted into useful products? So cholesterol is important for synthesis of vitamin D. It is important for synthesis of steroidal hormone like glucocorticoids, mineralocorticoids, estrogens, progesterone, testosterone, all male and female sex hormones. Then it remains in the form of cholesterol esters, and it is an important component of various plasma membranes also. Then bile salts. It is important for synthesis of bile salts. Bile salts are important for fat digestion in the intestine. Also, they are important for absorption of various fats and lipids from the intestine to lymph. Then it is also important for synthesis of 25 hydroxycholesterol, which is precursor for vitamin D. Then what are the this is the view of important uses of bile acid. Bile acid is important for emulsification of fats or lipids, digestion and absorption of fats, then absorption of fat soluble vitamin, prevention of cholesterol precipitation, and then elimination of cholesterol. So these are the various uses of uh, bile salts. Then cholesterol degradation and synthesis of bile acids and salts. So bile acids and bile salts are synthesized into the liver. From the liver, they are being collected into the in bile in the form of bile juice in the gallbladder, and whenever being required, they are being released into the intestine. So formation important role of liver is in synthesis of bile acids and bile salts, then secretion of bile salts in the bile, then finally excretion of bile in the intestine. So this is excreted in the intestine. Then there is an enterohepatic circulation of bile acids or salt. So not all of whatever amount of bile acids and bile salts is being excreted in the intestine, all of it is not being excreted out through the feces, but more than 70 to 80% of cholesterol, this amount is undergo enterohepatic circulation via the portal vein. It reaches again to the liver, and from liver it again comes into the gallbladder via bile, and then from bile bile reaches to the intestine. So this cycle goes and goes on. So this is known as enterohepatic circulation of bile acid and bile salts. Then finally all of these are excreted gradually in the form of feces. So consistency of bile contains 97 to 98% of water, 0.7% of bile salts, 0.2% of bilirubin, 0.51 percentage of fats, cholesterol, fatty acid, and lecithin, etc. are there. Then 200 milliequivalents of inorganic salts are there. About 400 to 800 ml of bile is produced per day in adult human beings. Then synthesis of bile acids. How it is happening? There are two types of bile acids. One is primary and secondary. So the endoplasmic reticulum associated cytochrome P450 enzymes are involved in the synthesis of this bile acid. So this you can see the cholesterol is here in the presence of NADPH, and the enzyme is 7 alpha hydroxylase in the presence of vitamin C, and cytochrome P7A1 is responsible for synthesis of bile acids. So in presence of all these ingredient, it converts up finally into the 7 alpha hydroxy cholesterol. Then further 7 hydroxycholesterol can be converted up into the primary bile salts. They are cholic acid and chenocholic acid. So both are 24 carbon containing, and the enzymes required for this conversion are 12 alpha hydroxylase enzyme, and NADPH is being required, oxygen is being required, coenzyme A is converted up into propionyl coenzyme A. So this is how the primary bile salts are synthesized. From primary bile salts, there can be synthesis of secondary bile salts. So it happens through liver peroxisomal enzyme conjugation reaction. So this cholic acid and chenocholic acid, um, from glycine and taurine, they converted it into glycocholic acid and taurocholic acid respectively, and which is in the bile. And from via the intestinal bacteria, they are finally converted up into the deoxycholic acid. Glycine and taurine are released, and this is how it is converted up into the deoxycholic acid with the help of intestinal bacteria. So this is an secondary biosol. Similarly, the chenodeoxycholic acid undergoing the same path, same steps, and finally converted up into the lithocholic acid. Then biochemical basis of use of cholestyramine is a drug. Which is drug? It is important in reduction of hypercholesterolemia. So how it happens? This is the actually by it is an bile acid sequestrant, that is a cholestyramine. It bile acid binds in the gut with this compound. It prevents their reabsorption into the blood, so promote or facilitate their excretion. It also leads to inhibition of bile acid synthesis in the liver, then thereby diverting additional cholesterol into the bile acid synthesis. So dietary fibers also bind bile acids and increase their excretion. So this is how we can download the concentration of cholesterol in the blood. Then lipid profile. What is a lipid profile? Lipid profile does consist of panel of certain lipid constituent present in the blood. That is, first is cholesterol, second is triglycerides, HDL, LDL, and cholesterol HDL ratio. So cholesterol, the desirable is it should be less than 200 milligrams per dl. The borderline is 200 to 239 milligrams per dl, and more than 240, the patient subject is at high risk of um, cardiac diseases. Then triglycerides concentration should be less than 150 milligrams per dl. So borderline is 150 to 199. More than 200 is always a risk. Then cholesterol, HDL cholesterol, a normal range is 60 milligrams per dl. Borderline is 35 to 45 milligrams dl because it is a good cholesterol. So its concentration, if it decreases, so it is again a risk for cardiac diseases. Then, so less than 35, it is very, very highly injurious condition, and that is a high risk. Then LDL cholesterol level should be in between 60 to 130 milligrams per dl, and 130 to 160 milligrams per dl is the borderline, and more than 160 milligrams per dl, it leads to high risk for cardiac diseases. Then another parameter, that is the cholesterol HDL ratio. And if the ratio desirable is 4, means cholesterol, if it is 200, HDL should be 60, so divided by it should be nearly 4. So 4 is the desirable ratio. Then borderline is 5. If it is more than 6, it shows the patient subject is at high risk of cardiac diseases or atherosclerotic event.